Good morning, everyone, and could be good night, a good day, depends where you are. Here we are again, Bioverse webinar episode number 6. First of all, happy Lunar New Year. I know a lot of people from Asia still celebrating the New Year. Some of them already started your day. Happy Lunar New Year, and for those loving birds, happy Valentine. But still we are working and wish you, I'm pretty sure you have some dinner plan already. Today is my great honor to have such a decorated group of panelists who have brought more than, I would say, based on my calculation, about 20 drugs to the finish line. And, I just cannot wait to listen to them and learn about in my opinion, one of the most critical steps for us to bring innovative drug to the patient and help the humanity. Before that, let me do a quick introduction of who we are in case this is the first time to listen to Bioverse webinar. All right, I believe everyone can see my screen now. Who we are, Bioverse webinar. This is a monthly show co-produced by Sino-American Pharmaceutical Professional Association with Philly chapter. We call that ourselves SAPA-GP in short, and in Science We Trust community. SAPA-GP is a nonprofit founded more than 20 years ago. Right now, more than 4,000 members, more than 160 sponsors, and over 1,300 LinkedIn followers. And in terms of in Science We Trust community, we are a lot younger, but we have been growing pretty fast. And the one thing we are most proud is we have vast majority of China biotechs with at least one C-level member exactly as our members. Today, really, I don't want to spend too much time talking about ourselves. Today, it's really my great honor to have this panel, like I mentioned, decorated drug developers. And if you look at our introduction, you will say they have spent more than 70 years, 7 decades in industry. I want to emphasize industry to bring innovative drug to the patients. And today, my role is really just, the MC, if you may, to introduce the webinar. But today, really, Li Yan, our moderator, gonna be really chair the whole session. So with that being said, I would like to hand this over to Li. Please take it from here.
Hey, thank you, Leo. And good morning, good evening, everyone. Happy Valentine's Day again. And for those in China, thank you for sacrificing your Valentine's Day and joining this episode. So as Leo said, we're going to discuss today is really what are the roles of a clinical and physician scientists in terms of developing drugs. And we have really 3 distinguished panelists. I thought 4 of us would add up to 100 years, but I guess we're still young. So 75 years of experience, so that's pretty amazing. And I noticed that we have 3 oncologists and 1 immunologist from a clinical perspective. And we spent time in biotech and a big pharma across the whole sector. So it's really interesting that today we're going to have this opportunity to discuss the topic. First, I would like to ask our panelists just to have a very short self-introduction, and we could start with the first name alphabetically. So Axel, if you could start.
Yes, first of all, thank you for having me. It's an honor to be here and an exciting program. So I look forward to this discussion. My name is Axel Hus. I am an entrepreneur, biopharma leader, and clinical drug developer for about 20 plus years. I think my most important contribution was the introduction of immuno-oncology about around 2011 with the approval of ipilimumab and a variety of other programs that have been developed that were quite innovative and different than what the standard at the time has been. So introducing innovation has always been something close to my heart, and I look forward to discuss this today.
Thank you, Axel. David, if you could start.
Hello, everyone. Thank you to Leo and Yi for the opportunity to participate. I'm very honored. I was a medical oncologist, first at Memorial Sloan Kettering Cancer Center, then at Dana-Farber Cancer Institute, where I saw patients and also ran a laboratory focused mostly on lymphoid malignancies. And I joined MSD Merck as the vice president for discovery oncology, overseeing the preclinical effort across all of oncology and hematology to develop new medications a little over 2 years ago. And thank you again.
Okay, David, I think I figured out you are the one who dragged down the average time we spend in industry. But if we add your career track tenure with academia and clinical, I mean, we would be hitting 100 years. So, Francisco.
Hi, good morning, good evening. Happy Lunar New Year. Gonshi Fakai. I am Francisco Leon, immunologist, drug developer. I have had a career in academia, big pharma. I actually was at BMS as well, working on ipilimumab in the very early days. That was such an incredible revolution that was taken forward by my colleagues. But I switched to immunology, and I've worked primarily on autoimmunity, bringing drugs forward. Eventually became an entrepreneur and started a couple companies that were acquired by big pharma, such as Cellimmune and Provention Bio, which was acquired last year by Sanofi. I'm currently the CEO of a small startup company called Tolerance Bio, and we work in the thymic space, thymic regeneration. We are raising a seed round, and there's still room for investors. So if anyone wants to chip in, just let me know after the call. Thank you.
Thank you, Francisco. So both you and Axel, I guess you touched the ipilimumab, and Axel, you took it to the finish line, got this approved. This is very innovative immuno-oncology. Really start the whole era of immuno-oncology therapy. So could you both just share your perspective in the very, very old days when immuno-oncology was not a hot area? And how did you come up with a clinical development plan with a goal of getting this innovative drug and a very new concept approved in the clinical and start a whole therapeutic area? Anything from clinical development plan, indication selection, sequence of indications, patient population, regulatory strategy, etc.
Well, I will just say that the one thing that is probably true for any drug development program, but particularly here, follow the science. Let the science drive it, because when you make a major paradigm shift, like from treating the cancer directly with a poison like chemotherapy and following all the principles that entails in clinical development, largely aiming to shrink the tumor and kill cells, and you switch that to not treating the tumor and actually, treating the immune system, therefore having an indirect mechanism that takes more time to see clinical effects, and then, make the right endpoint choices where it's actually no longer about just shrinking tumors, because conceptually you're recruiting T cells, you're recruiting an army of cells that travels to the tumor, and the tumor may not just shrink right away. It may increase in size first before it shrinks. And while that seems obvious, it's not obvious in the clinic when you actually have a patient in front of you and the tumor has just gotten bigger on a CAT scan. You might declare that a drug failure, right? So changing that view, changing the paradigm from what do we measure, why do we measure it, and how do we translate that into a successfully powered study that can report a meaningful outcome? That was difficult to do and took a lot of rethinking of the way we treated patients at the time. And so what it meant for us was not just follow the science, but also educate the community on a new way of looking at clinical development. So that's the reason why this name immuno-oncology exists. We invented it basically to illustrate that drug development in oncology using chemotherapy principles needs to be rethought if you bring the immune system into the picture. So immunology and oncology get blended into immuno-oncology. And then underneath that umbrella, we created all these other new tools like immune-related adverse events, which you now find as a separate category of adverse events in any drug label for an IO drug. Doing it the first time, though, was quite challenging because FDA had to buy in, and it was a long process to get this coordinated. And then beyond that, immune-related response is, what I just said earlier. Tumors can grow before they shrink, or that they may never shrink, but your patients may live longer by substantial margins. So you're creating the equilibrium here between the immune system and the tumor. It is a different way of looking at things, and that takes time to establish. And it is a convincing process of the scientific community, and especially the clinical community that deals with patients every day and needs to convince patients that they should stay on drug if things don't look conventional. So, I think the biggest observation that we made was if the tumor doesn't shrink, but you still have impact on the disease, you cannot use response as a primary endpoint for a trial. It may not give you the readout that you want. So the ipilimumab program, when it began, was started as a chemotherapy program. It was treated like a chemotherapy program because there was no paradigm to do this any differently. And we established a new paradigm, rethought all the basic principles, and then changed midstream in a blinded trial the registrational endpoint from response to survival, which made the trial successful. The trial failed on response, but it actually was successful with a wide margin on survival. It's just one simple illustration of following the science. So I stop here because we can spend the whole day talking about this one.
No, I mean, this is a really fascinating story, right? Changing the endpoint really based on the science, based on the clinical observations, and really adapted to what science tells you what's happening in the patient. So, Francisco,
yeah, I think I just want to continue that story and bring it back full circle now to immunology, because it's really interesting. As Axel said, it was very counterintuitive in the early days of checkpoint inhibitors, because if you recall, the checkpoint inhibitors started in immunology. We were working on co-stimulation. Before the word checkpoint inhibitor was coined, there was CD80, CD86, CTLA4, and the goal was to reduce the activation of T cells in autoimmune diseases. And the first drug was CTLA4 Ig, abatacept, which became ORENCIA, and then belatacept, which became NULOJIX. Those were the BMS drugs for autoimmunity, for transplantation. And then the oncologists said, if we follow the science and we have found a way to reduce the activation of T cells, why don't we turn it around and we use it to activate the T cells in cancer? And of course, that initially was a bit scary. Well, are we going to cause autoimmunity as a side effect? Of course you will, but you will cure people from cancer, so it's worth it. And from that initial discussion, with all the work done by Axel and his colleagues, and by David in the clinical setting, they were able to cure cancer, creating all this entirely new paradigm. And now we take it back into immunology. So all the checkpoint inhibitors induce, they release the brakes that keep T cells exhausted, right? So then the question is asked in the opposite way. So can we now induce exhaustion of autoreactive T cells in immunology, in autoimmunity? And that's how teplizumab came to be. Teplizumab, anti-CD3, now TZIELD, is a drug that induces exhaustion of autoreactive T cells. It's basically the other side of the coin. And again, it's the same principle. Let's follow the science, even if it's counterintuitive. And then let's develop the new clinical paradigm. So just like oncologists had to create an entirely new trial design, endpoints, regulatory process, the same had to happen to prevent autoimmunity with a an inducer of T cell exhaustion. So prevention and the organizations that came before prevention had to talk to the regulators about treating people who have subclinical autoimmunity, identify them with biomarkers, predict when they are going to become clinically autoimmune, and treat them before that happens. This was a bit difficult with regulators because they think, well, these are healthy people. Well, they are not. They have a subclinical autoimmune disease, and then developing those those biomarkers, those trial designs, etc., was critical to again advance the field, but always following the science, but then with improvement in clinical trial designs.
Great. And David, I would like to ask you from a medical oncologist perspective, and as a principal investigator perspective of clinical trials, would you please comment on IPI as well as other immuno-oncology perspective?
Well, I am perfectly willing to admit that I was one of the people who, before the data for ipilimumab, said this stuff is never going to work. And I could not have been more wrong, obviously. And it's ironic now, of course, that I'm an MSD. I think that what Francisco and Axel are really hitting on is the incredible value of having the ability to reverse translate, to make an observation in the clinic, and then, as Axel said, follow the science. That requires several things. First of all, it requires really careful observation of what's happening in the clinic. It requires commonly on-tumor biopsies or other approaches where you can understand the relationship between PK, PD, and efficacy. And even efficacy may have previously been defined as enlargement of the tumor, but as Axel described, the paradigm may be changing because that may not indicate actual tumor growth. It's simply instead infiltration with immune cells and inflammation. And then it requires having a preclinical organization that can take those observations and generate hypotheses and then try to validate approaches that can overcome or inform on those clinical observations. And then a clinical organization with the fortitude to be able to say, we're going to change midstream, or we're going to build the next compound that should be combined with this drug that will overcome resistance. And that overall really is the key to being effective. And even as Francisco said, taking an observation from one set of diseases and moving it to another set of diseases, there has to be capabilities at each stage, and there has to be an enormous amount of trust within the organization. Because as we all know, if the clinical people are not listening to the preclinical or vice versa and so on, none of this translates the way we want it to.
Yeah, and I add one more comment to this. You know, one of the biggest challenges that we had was not just the science, but it was also to get other stakeholders aligned with the process. If you imagine you have a trial that's planned for 3 years with a response rate endpoint, and you switch to survival, now it's a 6-year trial that costs substantially more, and somebody's got to fund this. How are you going to do this? Who do you need to convince? Do you actually keep going? While other programs, and I remind you of tremelimumab that ran parallel with ipilimumab, another CTLA-4 targeting antibody, which shut down the program early. So if you have all these things going on in parallel, kudos to BMS for finishing the trial and actually proving the point. It was really important. Follow the science and bring all the stakeholders along.
Yeah. So speaking of that, you know, if you did not change the endpoint in the midst of the trial, this could be read out as a failed trial, and the whole immuno-oncology era would be coming, right? So follow that topic, Francisco. I want to ask you, in terms of I would call salvage that antibody, right? And it was really from not only one failed trial, but multiple failed trials labeled as failed trials, including phase 3 trials. Could you tell us and share with us how did you, based on the science, rediscover and found the value of the drug and sold it to Sanofi for billions of dollars?
I think David made the critical point there. There's so many clinical trials that are labeled a failure, and they're not mined enough for lessons. And it's so difficult and so costly to do a clinical trial just to put the drug on the shelf at the end. So there are lots of opportunities for drug repositioning if people take the time to understand the trial, to look at the patients in subsets. Of course, the story of Glivec, right, that was rescued after a failed trial, but had worked in one patient or something like that. That's like the first example. So in the case of teplizumab, unfortunately, it's a story of a very lengthy development plan where the primary endpoint had always been successful in the phase 1, phase 2 trials. But when the sponsors, this was Macrogenics and Lilly, went to the FDA to conduct the phase 3 trial, the FDA forced their hand into a new endpoint, which had never been tested before. That is a very bad place to be, where you're testing an endpoint for the first time in phase 3, and that's your primary endpoint. So it's likely not to work, and that's what happened. They were also making a lot of changes from phase 2 to phase 3, and all we had to do was that reverse engineering to realize the drug actually worked, but it didn't work in all these new populations and this new endpoint. Let's go back. And we just repeated the study that had failed, but adapting it to what had worked, the population that had worked. Now, then we go lucky, and of course, the harder you work, the luckier you get, right? So we go lucky, and there was a small investigator-initiated study going on in parallel in the prevention of T1D. So while Prevention Bio worked in the treatment of T1D, the investigators worked in the prevention. And actually that trial read earlier and was the first approval. And that was what led, together with the promise of the second indication in treatment, that led to the acquisition. So also I want to throw a word for investigator-initiated studies, which are so important. They are creative and they can unleash the value of a drug.
That's great. That's great. Thank you so much, Francisco. So we all talked about trial design, follow the science, etc. But when the rubber hit the road, it's really execution is also equally, if not more important, right? So Axel, we'll come back to you because in your development of IPI, I mean, you have to come up with new endpoints. You have to change the trial design in the midway. You have to come up with a new assessment criteria for toxicity, etc. So from operational perspective, how do you make sure that when you have hundreds of sites over, all around the world, and they could actually come up and get you the robust data by the end of the trial that will get you the registration?
Yeah, so I don't want to make this just an IPI conversation, just to be more generic. There are some basic principles that applied at IPI, and they applied in any other program I've ever run. So in terms of successfully operationalizing a program, clinical development is a relationship business with many stakeholders, obviously, but primarily with the physicians that conduct your trial. I mean, we as drug developers working in industry, we're not really running the study ourselves, right? It is the investigators doing that. And being in close communication and having a trusting relationship with the key community that runs your trial, for me, has always been the centerpiece for everything that you do in drug development. And, if you have a doctor that sees a patient and the tumor has just grown, that doctor, in many cases, would just say, thank you very much, we're finished with this program. Or the patient stops treatment, we declare this a failure, and not open-minded towards any other interpretation. That can change if you actually have followed the science, you have good explanations, you have other clinical parameters than just tumor volume. Like we had patients that said, we feel a lot better. yes, that tumor looks bigger, but I feel better. There's something happening here that's positive. What do you do? As a physician, you need to take these things into consideration, be partner with the drug developer, and then get this to the next stage. And that's, I think, true for any drug development program. It was particularly important for ipilimumab, given the challenges that this program faced the very first time around. But it's true for any other program. Making sure drugs get developed in collaboration between all the key stakeholders. And then, of course, you go beyond the treating physician or the investigator to preferred partners, selecting the right CRO. In most cases, we in biotech, small companies, outsource a lot of the work that needs to be done. Having trusted partners there, having good geographic coverage for the different sites that you want to include, and then making sure that the studies are staged properly. So if we go back to phase 1, where the programs begin, you will not include everybody in phase 1, but you need to have a plan of how that unfolds as you go to phase 1B, phase 2, and then eventually registration programs. If you lay that out properly from the beginning, you can operationalize a successful program. But again, I come back to relationships are everything. This is a relationship business, and it's a collaboration between many stakeholders.
Great. David, I would like to come back to you as a PI from your perspective. What have you seen in your very distinguished career as a clinical PI? The do's and the don't do's from a sponsor perspective.
Well, first, it's important to clarify, most of my clinical trial participation was in ISTs and, really trying to drive translational science with the laboratory. I think I've gleaned a few insights. I could not agree with Axel more. It is all about relationships. And as always, it's all about incentives and disincentives. So, someone who is enrolling patients is not necessarily incentivized in the same way as someone who is really benefiting from the success of a trial. MSD made a decision several years ago during the development of pembrolizumab to try to build its own clinical trial organization rather than utilizing CROs. And I think that created incredible opportunity for employees of MSD who are incentivized really to drive the development of those therapeutics to work directly with investigators and treating clinicians at sites rather than having a CRO intermediary. That made a huge difference as far as we can tell, not only in being able to have those direct lines of communication, but also in building the relationships. So now there are relationships between MSD clinicians and those investigators at sites around the world. And it's just that much easier then to open up the next trial, to iterate on new concepts, and even to glean new ideas from those investigators, hear what they're seeing, what they think needs to be addressed next, what's the greatest unmet need, what are the possible combinations, and establish connections that we have found to be really invaluable.
Absolutely, David. I remember when we developed in Pembro, for the phase 1, we did numerous protocol amendments and enrolled hundreds of patients and got data supporting melanoma and non-small cell lung cancer indication approval. And it's impossible to walk through a CRO if you think about it, right? But not every company is as lucky as MSD's got, I would say, almost unlimited resources, right? So many of us now in small biotech companies, as Francisco, I want to come to you. I mean, when you were running Prevent Bio, it was a tiny company, and could you share with us how were you managed to do? I remember 6 clinical programs with such a small company, and how did you manage all the CRO services, etc.?
Yeah, so if we switch gears to small organizations, for those of you who may work in a small company or may start a small company, one of the things that has worked for me in the past is to keep the organization lean internally, not to overbuild the infrastructure, but to spend a lot of time on the road, building those relationships, and to employ very, very senior people. So both my companies, Cellimmune and Prevention, were virtual. We didn't have an office, but we only hired extremely senior drug developers who didn't need training. And we spent so much time on the road meeting each other and meeting with the sites, with the investigators. As David said, actually, the first drug that we licensed for prevention came out of a bar conversation. Teplizumab came to prevention because of the relationship between our chief medical officer, Dr. Ramos, and Dr. Jeff Bluestone. So at the end of the day, those relationships are extremely important. And then when you manage your clinical trials as a very small organization, it typically works best if you hire smaller organizations as well. So not the biggest CROs, but up-and-coming CROs, which you have to carefully vet, that are also mid-size or small, and they can give you a personalized treatment, and they care. They're vested because their success rides on the success of your trial. So that worked out for us.
Thank you, Francisco. So
Actually,
probably not every
before-
team will be
Sorry,
able
quick
to.
question. Can I jump a quick question?
Yeah, sure.
We're talking about relationship. So I found there's like, recurrent thing you guys have been talking about, how important relationship. But I think there's probably another side of the flip side of the coins. Like, I guess what you are really talking about is you have a good relationship. So my sense is you must have a lot of trust between you, the sponsor, or like, PI, which doesn't matter which side you are, and the other side. So my question for you is one step further. How did you build a trust which lead to the good relationship in the very beginning? What's a key factor? What's your checklist of building trust?
Well, you're now talking about interhuman relationships, right? This is not a clinical question at all. This is about, being open-minded towards the science, which unites us all, and the ability to really be available and communicate about difficult topics, right? It's not always about efficacy. In most cases, when you talk to a physician, it's about safety. And if you test a new drug, something happens that is unexpected, and you need to find a way to do the best for the patient in the study, and then eventually after that for the trial at large. And I have seen so many situations where one physician makes a negative observation in a patient that doesn't know the answer what to do with this. But you as the sponsor sit at the center, you have the experience of all the physicians in the trial. They may not talk to each other, but they talk to you. And you can share experiences from one physician to another and help in the particular situation that is a challenge. And that builds trust. It's really a communication process. So I just give you an example. If you, inflame the liver as a consequence of, trying to boost immunity against the tumor, and you basically send the patient into liver failure, how do you manage that problem? We have very little time, and the physician needs to be able to get an answer quickly. So the answer comes from, we have seen this before in the trial somewhere else, and I can help you with that answer. And here is the immunosuppressive medications and the dosage that you need to give to the patient to control this problem. And so you translate from one side to another, and the physicians will be grateful for that, and the patient, of course, will benefit from that. So we have had so many cases where that was done successfully, and that unites the entire community. So it's just one example, but it's a communication process.
Got it. Good point.
I would sort of take the obverse of that. I'd say every clinical investigator has had an experience where some company tried to whitewash data, and they knew exactly what was happening in their patient, but response, was counted when there wasn't really one, or a safety signal was not paid attention to or not adequately cataloged, and so on. And I think that just builds a certain wariness, right? Because if you're an investigator, you're worried about the least common denominator until you establish an experience with the company that you're working with and recognize that they're not the type of company that does this. So I think for a new biotech, it can be really challenging. There's importance clearly in picking CROs who have good reputations, who have established relationships with the investigators. And so there'll be less concern than if you're starting completely fresh.
Absolutely. And especially during the early stage of clinical trials when the PIs don't have a whole lot of experience, right? So that's even more important. So just be transparent and be comprehensive and be responsive. If you see something, pick up the phone. Don't wait until the emails. It might be too late. So we don't have a whole lot of time, but I want to ask a last question. Everybody, small biotech companies, startups, they're looking for exit, right? And it's hard for a small biotech company to take the drug all the way to the finish line to get approval. And even approval, you got different variety of challenges. So what we need to really do is to find the exit. And business development and working with big pharma appears to be an immediate and feasible and very helpful, very helpful exit. So Francisco, I want to ask you, because you've done it twice now, right? Exit and working with a big pharma. From your perspective, what are the key criteria driving successful business development besides hitting people at the bar and with trust? What else will be really the selling point? And then David will come back to you to comment from a big pharma perspective. What are you looking for?
Yeah, I think it's always about finding out what the stakeholder needs are. You have your own program, you have your own desire to help a group of people. And you have to find out who is also interested in helping the same group of people. It may be an R&D need, a group in R&D in a big pharma company that has a need in the pipeline for that same patient population, or it may be a commercial need. The sales force needs more drugs to sell because they are not busy enough. So you have to identify those alignments and then establish the relationship and work with them. In many cases, it could be a regional relationship, right? So China is a great place to in and out license drugs and to establish alliances. You have to try to understand how the market is in other places and the incidence, the prevalence of disease, the unmet need. So at the end of the day, yeah, just try to understand other people's needs and whether you can solve their problems with your solutions.
So I love Francisco's answer. I'll basically say the same thing in a slightly different way, which is every pharma company has its own culture, its own history, its own needs, its own portfolio, its own personalities. And all of those affect what it will be most interested in pursuing. And it's very dependent on what stage of development the asset is at. So a preclinical asset, I think a lot of it is about partnership. Are we not only going to acquire whatever chemistry and biology or whatever else has been done, but are there people in the company who we can partner with if we license or we co-develop and so on, and have, again, a trusting and really fruitful scientific relationship where we can not just develop this asset, but we can learn internally and make ourselves better. If it's a clinical asset, then I think it's a lot of it is about POS and NPV calculations and how well it fits into the portfolio. I do think there are some things that come up when we dig really deep into these assets. The drug can look fantastic in phase 1, but if it's all just investigator-read CTs and then we do our own read of CTs, obviously the overall response rates have to hold up. We have to be really confident in the data that's available. And then something we frequently raise is manufacturing. If this asset is nearing the end of phase 1, getting into phase 2, and we recognize that it doesn't meet certain standards, MSD holds very high standards for manufacturing, that means going back, for example, for a biologic to cloning to a single cell line and so on. And that's going to delay the phase 3 by a year or 2 or even more. All of those are really important considerations. So I think as a biotech thinking about pitching to a pharma, not only what is it that you're hoping to get, but what is it that you're really offering, and how does that entire package fit with the company's needs?
I have a quick
both
question.
Small biotech and large pharma. Any comments from you?
Yeah,
Excellent.
so I will just say, you know, everything that was said makes sense, is right on point. And it becomes this, you know, usually balance between business incentives that you always have to consider. And the biotech view there is quite different than the pharma view. And then, of course, the clinical incentives that the teams that want to work on these assets have from an R&D perspective. So we covered both of those in some ways. But if I think about it from the biotech perspective, you know, it depends on what the asset is in your own portfolio. If you have a portfolio, if you're not a single asset company, that means if you want to partner your lead asset, that has significant implications for the valuation of the company and is commonly not desired from the biotech perspective. If you talk about second, third, or fourth assets in the pipeline, co-development partnerships are quite attractive because you can share cost, you can share, you know, operational challenges, you can have wider geographic reach between two partners instead of doing it yourself, and you can extend runway that way as well. So there are lots of, you know, components that have to play into this that are way beyond just clinical development. They're business considerations, and they're different between pharma and biotech. You know, having run a large portfolio at GSK at the time, I also see the pharma perspective, and it is really then a question of capital allocation, strategy, and portfolio prioritization. If you bring a new asset into that portfolio, and the priorities here might be quite different than the ones that a biotech company might have. So finding the balance, understanding the strategic needs of your potential partner is essential to make this a story. And I think we heard that from the other two, but you know, I just give you a slightly different angle on it.
Great, thank you. I think we're running out of time based on Leon's agenda. So Leon, well, first I want to thank all three of you, Axel, David, and Francisco, and back to you, Leon.
Yeah, so there's one question actually from the audience. I think actually this is probably really relevant. I would probably extend the webinar by another 5 minutes because it's very important. The question from the audience really is, what's your experience with the clinical trial development in China? Any advice do you have for Chinese biotech companies in terms of the assays, how they develop the drugs over there? I mean, especially for David, right? You have seen a lot of assay. I'm pretty sure quite a few of them came from China, and you trust some of them, like Colon, right? You license quite a few of them. So if you like, you know, put your lens, you know, specifically for China biotech, give you a little bit of advice and comments on that, I would be highly appreciated for the audience.
Sure. Well, I think the ecosystem in China is just incredible, and the ability to innovate and to very rapidly drive composition of matter to the clinic, and then get early clinical signals with both biologic and small molecules. We would be absolute fools to ignore that or not want to really dive into the opportunities. We have made multiple relationships with Chinese biotechs, and Kelun Plus, as Leon mentioned, I think is the best example. We started with one asset deal, then we moved on to a second one, and then based on just continuing success, we expanded that into a very large multi-asset deal. And I think both sides are really reaping the benefits there. We're looking at multiple biotechs right now. The challenges include the issues I mentioned around manufacturing and just ensuring that there's manufacturing that matches what MSD does. Some of the restrictions in China, I think that you all know about exportation of data and certainly samples. Those are concerns, I think, especially in the very early space, or if there's intense reverse translation that we want to try to organize during the early clinical development. Those are not insurmountable challenges, especially for a company like MSD. We have an MSD China a very large organization that can help to buffer some of these challenges, but they're not completely ignorable either. So I think we and other large pharma ex-China are thinking as strategically as we can about how to take advantage of that incredible ecosystem.
Yeah. And if I can add one comment here, you know, we said as a guiding principle for a good clinical development plan, you need to follow the science. I think we all agreed on that. Now, another thing that's important is to think globally. Think when you start the program and you actually want to have a presence in multiple geographies, including China, or even if you are in China and you look outward to a potential global development program, think about that right from the beginning in phase 1, so you overcome some of the obvious problems that will pop up when you just want to take the next step and you're missing something. You know, if you think globally, you can avoid many of those problems. I know this is a generic statement, but it is very important. I've seen it so often that a program begins in one part of the world and is still not considering anything else. And when you have data and you want to take the next step, you're not ready for it. So that would be, I think, an essential piece. Consider this to be a global program from day one.
And that's point. Francisco, anything you want to add before we conclude?
Don't forget the personal relationships. Don't forget the idiosyncrasies of the countries you work on. China has extraordinary capabilities. You have to go there. You have to meet people. It was difficult during COVID, but hopefully in the coming years, we can go back and meet each other often. So I hope that we strengthen our network and our links.
Fantastic. I think that's a perfect closing remark. Really boil down to a good relationship. This is a really global business, and we need to get talents everywhere from the world, work together to find innovative drug to help patients everywhere. With that being said, I would like to give a little bit kind of shout out, you know, a natural transition to, since we're end with the webinar with the China Biotech, I want to give a little bit promotion of 2 events. The first one, actually, Axel and I gonna be co-chair of events and IO360. If you are interested in immuno-oncology, which we talked a lot today, as you can tell, we welcome everyone to join us. Gonna be in Brooklyn, lovely place. And you can scan the QR code on the left to join the conversation. And we have some of the best speakers, which, you know, Axel can, you know, definitely, agree from the immuno-oncology space. That's one. Another one, since Yufeng is here, we have the incoming SAPA GP annual conference in 3 weeks, I believe. And we also have some session talk about, business side, along with many other stuff in this annual conference. So I hope I stop here for probably 30 seconds for the audience who are interested. You can scan the 2 barcode and join us. And we're looking forward to meeting you either in Brooklyn or Philadelphia. I believe it's in the King of Prussia, right? Over there. With that being said, thank you very much, everyone, especially those from China. I know it's pretty late already, but anyone who attended today's webinar, I hope you have got a little bit smarter by listening to those panel of decorated drug developers and physician scientists. And with that being said, we wish you the very happy Valentine's Day and Lunar New Year. And we'll see you again next time very soon. Take care. Bye for now.
All right,
Thanks,
take care.
everyone.
Bye-bye.
Thank you.
Bye-bye.